osmosis? Osmosis means movement or diffusion of water across the semi-permeable membrane from a region of its high concentration to its region of low concentration until the equilibrium is achieved. Main is the equilibrium. Okay. Now the net rate and direction of osmosis, what does it depend on? The pressure gradient as well as the concentration gradient. And there is an experiment which is going to demonstrate that how osmosis takes place. What are we doing in this experiment is, first of all, we are covering the thistle funnel. We are using a thistle funnel and we are covering its mouth with a semi-permeable membrane. So this is a thistle funnel that I am using in this experiment, and I am covering its mouth with a semi-permeable membrane. So this is how it is covered. Okay. And it is filled with sugar solution and kept inverted in a beaker containing water. So what are we putting in is sugar solution. This is sugar. So we have filled it with sugar solution and inverted. This is the way it is inverted in a beaker containing water. Now this is a beaker which contains water. Clear? So let's mark water in a beaker, and this is the sucrose solution. This is the membrane with which it is tied. Thistle funnel is tied, right? And here we mark the levels. Okay, the level. Now what happens here is, numerically, what are we calculating? The osmotic pressure. What is that osmotic pressure? Osmotic pressure is equivalent to the osmotic potential. Osmotic potential. But you know, the sign is always opposite. Osmotic potential is the solute potential, but the sign is opposite. Keep in mind. Now let's begin with the experiment. The mouth of the thistle funnel is tied with a semi-permeable membrane. It is filled with sugar solution and kept inverted in a beaker containing water. Water will continue to move across the membrane, and hence the solution in the funnel will also rise. Right? It will also rise. Now if pressure is applied on the solution from the upper part of the funnel, this movement of water will be further stopped. Clear? If I am applying pressure to the solution from the upper part of the funnel, the movement of water, it will be stopped. Clear? Here, what are we calculating? That on applying pressure, there we get a final level, and this was the initial level, and as the pressure was applied, the movement stopped. It is equivalent to that osmotic potential, that is solute potential, was giving us the value of osmotic pressure, and osmotic pressure is equivalent to osmotic potential or the solute potential, but the sign will be opposite. This is what is applied from here, and the pressure is applied from the top. Okay. Next is plasmolysis. What is plasmolysis? Taking up this only, the movement of water in and out of the plant cell or in and out of the through this membrane depends on the nature of the external solution. If the external solution is said to be isotonic, means it will balance the osmotic pressure. That is, it has the same solute concentration as the cytoplasm. Then automatically the flow will remain constant, and it has the same it has the weaker solution than the cytoplasm. It is said to be hypotonic, means it will have a higher water potential than the cell. And in case the external medium is more concentrated, then it has a lower water potential than the cell. Okay, let's write them down. It is the movement of water in and out of the plant cell. Now the first condition is, if the external medium or external solution, let's say external solution only, external solution, it is isotonic. Okay, imagine these two situations only. If the external solution is isotonic, it will balance the osmotic pressure. It will balance the osmotic pressure. Therefore, it has the same solute concentration as that of cytoplasm. So, same solute concentration as that of cytoplasm. If, second condition, the external solution is weaker than cytoplasm, it means it is hypotonic. Then such a solution has higher water potential than the cell. Okay, we are imagining this as a cell. So what will happen? This solution has higher water potential compared to what is present inside. Obviously, because it is hypotonic. Now if this is more concentrated compared to this, means अब यहाँ sugar solution हो और यहाँ water हो. Okay, then we call it hypertonic, and it has a, when it is hypertonic, then it has a lower water potential than the cell. Clear? Now I'm writing here. I'm not removing this diagram because it will help us to understand the things more clearly. If the external solution, it is hypertonic, तो हमने इसको क्या लिखना है? वीकर की जगह स्ट्रॉन्ग. इफ इट इज मोर स्ट्रॉन्ग, ठीक है, मोर कॉन्सेंट्रेटेड, then automatically it has a lower water potential than compared to the cell. Now we have got three conditions. We have a hypertonic solution, we have a hypotonic solution, and we have an isotonic solution. If we are keeping the external source, that is the plant cell, if we are placing it in a hypertonic solution, obviously the solution exerts a pressure. Let's keep a plant cell also. Okay, and this is the vacuole of a plant cell. Let's draw it like this for more clarity. We say if the plant cell is placed in a hypertonic solution, the solution exerts a pressure higher than the osmotic pressure and water moves out of the cell. Okay, this is hypertonic. Here the solution is more dilute compared to this condition. यहाँ पे ज़्यादा concentrated है, तो क्या होगा? सेल में से हमेशा सॉल्यूट बाहर आएगा. सो कंडीशन विल बी हाइपरटोनिक सॉल्यूशन में सॉल्यूशन exerts more pressure higher than the osmotic pressure. and ultimately what will it result into? Exosmosis. Exosmosis होगा. Okay, and if exosmosis will take place, it will result in the shrinkage of the protoplasm of the cell, vacuole and the protoplasm, which recedes the cell from the cell wall, and the phenomena is plasmolysis. So what do we observe in hypertonic solution? It's plasmolysis, and the cell becomes flaccid. Now if this freshly plasmolysed cell is kept in a hypotonic solution, okay, where hypotonic, dilute concentration of water, and we know by now the cell is shrink. It is completely shrunk under the given conditions. Here the vacuole has receded to this limit. Now water will enter the cell, okay, and a hydrostatic pressure develops inside the cell. A hydrostatic pressure will develop inside the cell and the cell regains its turgidity. This is called deplasmolysis. Okay, cell will become turgid as it will gain water. And always remember, a plasmolysed cell will show deplasmolysis. Clear? A plasmolysed cell will show deplasmolysis. So first of all, we have to show that it is first kept in a hypertonic solution. On effect of that, when the cell will shrink, then it is shifted into or kept into a water solution or a hypotonic solution. Water will start entering into the cell, hydrostatic pressure will develop, cell will regain its turgidity, and after plasmolysis, deplasmolysis will take place. Clear? Moving to the next concept, that is imbibition. Imbibition is a special type of diffusion. Here what happens? The water is absorbed. Absorbed in such a manner that it is called adsorbed by the solids, colloids. Because why adsorbed? Because it is absorbing in a manner that not making it as a solution after absorbing water, right? Like kneading the flour. जब मम्मा आटा गूंथती है तो क्या होता है? It absorbs the entire water, that is adsorption. It is causing the solids and the colloids to swell up in volume and no solution is formed. Here we say jamming of the doors during the rainy season. Wooden doors. Okay, kneading of the flour. This is an example of imbibition. So generally where have we observed it? We have observed it in dry plant material or seeds that act as an absorbent to imbibe water. Water is imbibed and swelling takes place. Here the water is absorbed in between the layers of the cells. So water is adsorbed automatically without forming a solution. Generally it swells up. Clear? Then when imbibition pressure, which is responsible for emerging of the seedlings, when the seedlings generally emerge out of the soil and establish themselves, it is basically the imbibition pressure which helps in. Okay, so it is imbibition pressure. Its significance is that it helps in the responsible for the seedling to emerge out of the soil. Clear? So obviously if this condition is applied, there are further necessary conditions which are to be followed for imbibition to take place. What is that? Number one, water potential gradient. Water potential gradient. It should be there between the surface of the adsorbent and the liquid. So surface of the adsorbent and the liquid or the water which is imbibed. Number two, affinity. Affinity between the adsorbent and the liquid which is to be absorbed. Affinity is the interaction between the adsorbent and the liquid which is to be imbibed. So this is how we show that osmosis takes place.